Good evening, Vanchika. Good evening, Amita. Okay, so I'm Rahul Saha, and today we'll be starting the male reproductive tract of the pertaining to the chapter the human reproductive system. Okay, so let's start with our male reproductive tract. So when we talk about the male reproductive tract, the first thing that comes to the mind are the testes, right? So let's get down to the function anatomy of the testis. Now, if we talk about the testis, okay, so this is plural, T-A-E-T-E-S-T-I-S is this singular, right? Now, each testis, if I talk about each testis is oval in structure, okay? It has oval structure and it has a length of about 1.5 inches and width of about 1 inches, okay? Length 1.5 inches, width about an inch, okay? That is the, you can say, dimensions of a testis. On the outside of each testis is a shiny covering of serous membrane known as tunica vaginalis, okay? Tunica vaginalis, which is the cover of serous membrane. Now, when we talk about serous membrane is the fold of the, you can say, visceral peritoneal membrane. That is the serous membrane. If I type here tunica vaginalis for you, tunica vaginalis. So I can see the image that it is put inside or it is covered by, you can say, the visceral peritoneal membrane. That is the meaning of tunica vaginalis. Okay, just look at this image. Okay, so this is your tunica vaginalis. Okay, this specific image is your tunica vaginalis. Good evening, Tarun. Good evening, Ayman. So this is your tunica vaginalis. This is the parietal visceral peritoneal membrane. Okay, and if you haven't learned about peritoneum, or if you want to just check the image of a peritoneal cavity and all, so I'll just show you the mesentery image. Okay. In class 11th, you may have learned, but I don't think you learned it in detail, the anatomy of the mesentery. Okay, this is how a mesentery looks. Okay, these are the folds of the peritoneum. So this is your parietal peritoneum. The inner fold, the same parietal peritoneum when it folds inside or towards the inside of the thing or inside of the organs, they are known as towards the inner side. So its same fold towards the inner side is known as the visceral peritoneum, and this is the, you can say, liver, this is the stomach, which are enclosed by the folds of the visceral peritoneum. Okay, so this anatomy, so when we will meet offline where I'll teach you this anatomy also later on, just before you write your NEET examination, because this is a very significant anatomy. Like various organs, they say that some are retroperitoneal, some are intraperitoneal. Means if you are completely, if that organ is enclosed by the peritoneal cavity, that is known as an intraperitoneal. But kidney and all, they will present here. So only they are covered by one layer of, you can say, the visceral peritoneum. The other layer they are not covered by, the other side is exposed. They are somewhat here. Okay, so the kidney is a retroperitoneal organ. Okay, that is also a neat question. Okay, so coming back to our discussion. So in the end, the thing is, the outside of each testis there is a shiny covering of a serous membrane known as tunica vaginalis. Okay, immediately under the tunica vaginalis is a thin dense covering of the testis itself, okay, that is known as tunica albuginea, okay. Then another one you have tunica, you can say, just open the slide code again. That is known as tunica albuginea. So we have tunica vaginalis, then we have tunica albuginea. Now inside each testis, okay, this is a dense covering of the testis itself, okay. This is the dense covering of the testis itself, okay, tunica vaginalis, then tunica albuginea. Now inside each testis are 250 compartments present. Those 250 compartments are known as testicular lobules, okay. Inside each testis, how many compartments are present? 250 compartments. They are known by the name as, or referred to as, testicular lobules, right? Now, each lobule contains, you can say, 1 to 3 highly coiled seminiferous tubules. Contains 1 to 3 highly coiled seminiferous tubules, okay, and each testicular lobules are separated from each other by specific tissue barriers, okay. Good evening, Jyoti. By specific tissue barriers known as septa. So from next week onwards, we'll be beginning our, you can say, discussion classes, revision classes in the offline manner. Okay, from next week onwards, from the new year itself. Okay, so you can send any of your doubts to me. You can send it priorly on my WhatsApp number. I'll be discussing them in the class, and for offline students, I'll be discussing them at that time only. Okay. So Vanshika and all, if you have any kind of doubts in any topic, okay, who are not able to come for offline classes, you can just text me your doubt, okay, just 1 day prior or 2 day prior or whatever, or 1 hour priorly, then I'll discuss your doubt along with the doubts of the offline classes as well, okay. So from next week we are beginning the biology offline classes in the center as only, okay, and biotechnology class will continue online on the Saturdays, okay. So the thing is, now there are specific tissue barriers known as septa which separate each testicular lobule, and each lobule contains 1 to 3 highly coiled seminiferous tubules, okay. The seminiferous tubules you can just compare with the renal tubules that we have learned. In case of class 11th, we have learned that the renal tubules are present in each, you can say, nephron. So the septa, they divide, uh, they demarcate each lobule. Okay. And now we know that the seminiferous tubule, if we talk about, are highly coiled structures. Highly coiled structures. Highly coiled structures are what? Highly coiled structures, you can compare them with the renal tubule. You had proximal convoluted, then loop of Henle, then distal convoluted tubules. Renal tubule is part of the nephron, right? The tubular part. So you can compare them with the tubular part, not the renal corpuscle part, the renal tubule part of the nephron, right? Now, if a single seminiferous tubule were stretched to its maximal length, okay, it would measure about 1 to 3 feet, okay. If one, that's the amazement, if one seminiferous tubule is stretched, just imagine if that is stretched to its maximal length, okay, that would be around 1 to 3 feet, okay. And then if all the tubules can be stretched simultaneously, that would be they will be covering a distance would be longer than a football field itself, okay. Just imagine. And again I will be relating this to the length of the renal tubules. If you elongate all the renal tubules, because 1.25 million nephrons are present in the renal tubules, if you elongate all the renal tubules with PCT, low FL and DCT combined, then you will get from one kidney itself, you will get a total length of about 141 kilometers. Just imagine how enthralling that is. 141 kilometers of renal tubules are present in each kidney. Okay, and one football length of seminiferous tubules are present in each, you can say, testis. And where are sperms produced? Sperms are produced in the seminiferous tubules itself, right? Sperms are produced in the seminiferous tubules itself, right? Now, we know that each seminiferal tubule contains male germ cells in various stages of differentiation. Okay. Now, we also know that each testis is suspended from the body wall by something known as spermatic cord, which penetrates into the pelvic cavity through the inguinal canal. I'll just show you the spermatic cord. You might not have heard about it. Okay, I'll just show you the spermatic cord. You'll be amazed. This is only present in males, right? The spermatic cord. Male and female anatomy difference, if you see. So this is your spermatic cord. If you just check out. Okay. Just look at this one. Okay. You have the pubic bone, you have the suspensory ligament of the penis. These are not real ligaments, these are just the folds of the visceral peritoneum. Okay, these are just the folds of the visceral peritoneum. Okay. So here what you have is, you can see here glans penis and then vas deferens. Okay. So I don't think this one has, this will be having, I think, the spermatic cord. Yeah, this one is better. This one I was talking about. This one, just see here. This is the spermatic cord. Okay, this one is the spermatic cord which penetrates into the pelvic cavity. You can see this one is penetrating through the pelvic cavity. Okay, through the canal which is known as inguinal canal. Okay, inguinal canal means the route through which the testis originally descended into the scrotum from the pelvic cavity before birth, because just before birth it gets descended, right? It is not descended, it is not developed outside in the scrotum, right? It is developed in the body. Then it descends. Okay, same thing happens in dogs and all. So then it descends. The actual development happens inside the body itself. So this is the inguinal canal that you are able to see, right? The spermatic cord brings it through the inguinal canal. Okay, these are the oblique muscles are there. Okay, and then it descends. This is the pathway of its descension into the, or its descent into the, you can say, scrotum, which is outside the body cavity, right? So each testis is suspended from the body wall by a spermatic cord. Okay, so by proper anatomy, each testis, that inguinal canal is all is present in the males, right? Is present or is suspended from the body, suspended from the body into the pelvic cavity. Into the pelvic cavity through the inguinal canal. Okay. Inguinal canal is what? It is the route through which the testis originally descended. The route through which testis originally descended to the or into the scrotum from the pelvic cavity. Into the scrotum from the pelvic cavity before birth. Okay, so that is what happens, right? Now each spermatic cord contains one of the accessory ducts, right, known as the vas deferens, a testicular nerve, and three coiled blood vessels known as the testicular artery, or you can say the spermatic artery, which carries blood to the testis, and epididymis, and it also contains two testicular veins known as spermatic veins, which carries blood away from the testis. Okay, so that is what the spermatic cord can contain. So just understand the anatomy. So this is your spermatic cord, you can say. So you can see the vast difference is present in the spermatic cord itself. Just look here. Okay, it is present in the spermatic cord itself. Okay, so each spermatic cord, okay, it contains the accessory ducts like the vas deferens, each spermatic cord contains the accessory ducts like the vas deferens. Okay. It does contain a testicular nerve, the autonomic nervous system, a testicular nerve. Then it also contains three coiled blood vessels, the testicular artery, also known as spermatic artery. Okay. Then you can say two testicular veins or spermatic veins. So 2 plus 1, 3. Two testicular or spermatic veins. So total three, you can say, blood vessels are contained. Okay. Now you can see the vascular system which serves the testis. It helps to maintain the testicular temperature sufficiently below, we all know, which is below the core body temperature. Why? In order to support spermatogenesis. Okay. Now the spermatic veins, they do what? They return the cooled blood from the testis branch into the, you can say, another network which surrounds the testicular artery. Okay. So this network is known as an anastomosis. Now what are anastomosis? If you have learned in class 11th in the blood chapter, anastomosis are nothing but if you have three or four arteries, okay, which provide, you can say, the same, you can say, organ, blood, that is known as anastomosis. I'll just show you. These are some technical, you can say, anatomical terms. That is why class 11th is very, very essential. You can just see anastomosis here. Just look here. So, so here it is surgical joining, but when it comes to the arteries, okay. So wait. Arteries in the heart. I'll show you that. If you have learned the heart chapter, you will be knowing this one. Okay. Just look here. So you can see here there are various arterial branches, okay, which provide blood to the same region. Look here, okay. This is the left main coronary artery, okay, that divides to become the LAD, left anterior descending artery, okay, and this is the left circumflex artery, okay, and then it will divide to become, okay, you can say, the later on the LAD will become D1 and D2, that is the diagonal artery, and later on this will become obtuse marginal 1, obtuse marginal 2, and also you can see these arteries, okay, this is marginal, here it is known as obtuse marginal, okay. So these arteries provide blood to this almost the same region of the heart, okay. Why is that done by the nature? Suppose you have a blockage here somewhere. So if this is providing the blood there, this is providing the blood there, so what will happen is the propensity or the intensity of the myocardial infarction wouldn't be that great because you're not cutting blood supply completely. Suppose four arteries are providing blood almost to the same region. So if one artery is blocked, still you are getting blood from the three arteries, right? So that is known as anastomosis. That is a way of evolution to protect, you can say, the specific organs. In same way, in case of our brain, we have this circle of Willis, which is a kind of anastomosis again in case of the human brain, which provides blood to the human brain through the carotid artery branches, right? So that is the meaning of anastomosis. So same kind of anastomosis are present in the, you can say, testicular network also. Okay. So this surrounds the testicular artery and this acts as a counter current heat exchange system, means cooling incoming blood in the adjacent arteries, okay, and then taking the cooled blood and then reheating it when it goes into the body. Okay, so that is the counter current mechanism that you have studied in case of the renal system, okay, in case of the kidneys, where they keep the water absorption, okay, they keep on absorbing more and more water and the Na plus Cl minus via counter current exchange and counter current multiplication. This is the counter current, you can see, heat exchange system as well, okay. That is how you can say the temperature of the blood coming onto the testis is maintained at 2 to 3 degrees lower in temperature, right? Now coming on to the seminiferous tubules. Now each seminiferous tubule is lined on its outside. You can say each seminiferous tubule we talk about seminiferous tubules. Now each seminiferous tubule, each one is lined, you can say, on its inside by the seminiferous epithelium. Okay, the tubule, the inside of the tubule, you can say, will have the epithelium cells. Okay, this is known as the same as the endothelial cells. So these are the seminiferous epithelium. And now the seminiferous epithelial cell, okay, are of, you can say, two types. We have the male germ cells and we have the Sertoli cells as well. Okay, we have the male germ cells and we have the Sertoli cells. Now, if we look at spermatogenesis or spermiogenesis, which we have already talked about, okay, so that occurs by the differentiation of type A spermatogonia into type B, and then type B goes through, you can say, the meiotic division, right? So that we are not going to talk about here, but one thing I'll tell you, that the immature germ cells, where do they lie? They lie next to the basement membrane of each seminiferous tubule. Okay, so you can say that the next to the basement membrane of each seminiferous tubules they lie, and these cells they multiply by meiosis, right? So we have talked about this specific, you can say, division leading to the formation of spermatids and then the spermatozoon. So that we have talked about. So there we will not be diverging our attention. So what we'll be talking about is the function of Sertoli cells. So Sertoli cells, as the function of the Sertoli cells we know. So we have already known that the plasma membrane of the Sertoli cells, okay, so what they do is they adjust to accommodate the gradual movement of the germ cells, okay, and thus the complex shape of the Sertoli cell is constantly keeps on changing. Okay, now sperm heads are released, okay, from the Sertoli cells at the point or at the event of spermiation, but additional functions are what? Additional functions of Sertoli cells are the secretion of testicular fluid into the tubular cavity. Okay, that is one additional function of Sertoli cells. We talk about Sertoli cells. Secretion of testicular fluid into the, you can say, tubular cavity. Next function is very enthralling. It can function as macrophages, means it can phagocyte, or you can say it can phagocytose or engulf the remaining of the degenerated germ cells. Okay, it can phagocytose the remaining germ cells which have got degenerated, okay, in the process, the remaining degenerated or dead necrosed germ cells. What else it can do? I had told you in one of the classes that the tight junctions between the basal portions of the adjacent Sertoli cells can also provide a very great barrier known as a blood testis barrier. Okay, so you can write it again, that blood testis barrier, okay, which is extremely necessary so that the T helper cells, okay, they are in the blood, so the blood shouldn't be reaching the sperms, because sperms, they have been produced by meiosis, meaning crossing over. So it may contain some, you can say, foreign proteins because of homologous recombination, and that will be detected as foreign, and there will be some immune response modulation or offense against the sperms, which will lead to sperm death. So you need to maintain the blood testis barrier. Same way, the blood embryo barrier is maintained by the decidual cells, okay, in the embryo, right? Now, when you can say blood testis barrier is maintained, okay, in addition to the blood testis barrier, you can say the blood testis barrier, what it does, it protects from the immune attack, that is fine. Now Sertoli cells also secrete various proteins, okay, including androgen binding protein and various hormones that we have already talked about, like inhibin and anti-mullerian hormone, okay. So androgen binding protein, then inhibin, okay, then anti-mullerian hormone. So all these specific hormones are also secreted. These proteins are also secreted by Sertoli cells. Now these cells also produce enzymes that convert testosterone to estrogen, okay, and also it converts estrogen to 5 alpha dihydrotestosterone DHT, okay. So they also produce enzymes, okay, which can convert testosterone to estrogen, like aromatase, like, right, to estrogen, and estrogen to another specific compound known as 5 alpha dihydrotestosterone. Testosterone, known as DHT, a very important compound for competitive examination. Now, Sertoli cells may also play an important role in the hormonal control of spermatogenesis. Okay, so because of the dependence of male germ cell development of on the, you can say, Sertoli cells, the rate of a man's sperm production is directly related to what? It is related to the number of Sertoli cells in his testis. If the number of Sertoli cells are very low, his sperm count will be low. So number of healthy Sertoli cells is proportional to the number of sperms produced, you can say. That is a general rule or a golden rule in case of sperm production by any man. Okay. Now the outer or the basal surface of the seminiferous tubules, okay, they are covered by a collagenous basement membrane. Okay, they are and it is thought that this basal membrane, what it does is it contains some specific cells, okay. So we know that basal membrane is present. Why? Because suppose this is the, you can say, seminiferous tubule, so here you will be having a layer of, you can say, basement membrane, because this basement membrane will connect this seminiferous tubule. Epithelial cells are present here, so it will be connecting these cells to the connective tissue cells here. That is the role of, you can say, basement membrane. Okay. So scientists have found that these basement membranes or the basement membrane contains some scattered myoid cells. Here some scattered, you can say, myoid cells are present in this basal membrane. It is thought that these muscle-like cells, myoid means myocyte type, muscle-like cells, they contract in on weekly basis to help move sperm cells and fluid through these seminiferous tubules. Okay, that is again one of the things of research, because how does the sperm move? Because they haven't been able to develop the, you can say, functional flagella in the seminiferous tubules. So how do they move? So the hypothesis is they are muscle type cells and they contract, and then the sperm cells, you can say, it will move in certain direction. Okay, that is the hypothesis of the movement behind the sperm cells which are present in the seminiferous tubule. Okay, now if we talk about the testicular tissue, okay, if you just go through a cross section of a testis, okay, so it does show a lot of small arteries, capillaries, small veins which are present in the interstitial, you can say, space in the testis. Now this is where the blood-borne products like oxygen, glucose leave the tissue and diffuse into the tubules. Okay. Now these products are extremely important from the germ cells. Why? Because there are no blood vessels within the tubules themselves. Very, very important. Okay, to know. So these intricate details you may not be knowing. So the thing is, if you go for a cross section of a testis, okay, so what you have is, we have closely packed seminiferous tubules and also, you can say, tissues occupying the spaces between these tubules. Okay, now these regions, the or the spaces which are present between the tubules are known as interstitial spaces. Okay, the spaces between seminiferous tubules, seminiferous tubules cluster, we have many clusters of seminiferous tubules, they are known as interstitial spaces. Okay, interstitial spaces. Now what happens is, there are these regions, they contain, or they are very, very specific for, or they are very, very vital to testicular function, because they contain small arteries, capillaries and veins. Okay, small arteries, capillaries and veins, they are present, right? Now this is where the blood-borne products like oxygen, glucose, they leave the blood, and just by diffusion they get inside the seminiferous tubules. So here you can say oxygen, glucose, they leave the blood and diffuse into the tubules. Why? Because these tubules, the tubules, they lack blood vessels. There are no capillaries, no blood vessels providing direct supply of oxygen and nutrient to them, right? There are no blood vessels within these tubules. So likewise, the hormones can also leave the interstitial blood vessels and enter the seminiferous tubules through the same basal membrane of the tubules. The basal membrane we have already seen, right? This is the, you can say, the basal membrane. So from here there are specific gap junctions through which they'll be able to reach the seminiferous tubules. Okay, so hormones and all, and oxygen, and you can say glucose, they all reach the seminiferous tubules through the basal membrane itself. Okay. Now this basal membrane, you will be asking a question that now the small molecules are coming through. Now what I said was this basal membrane, it also acts as a blood testis barrier. So what kind of barrier am I talking about? The thing is, understand it carefully. This basal membrane and the blood testis barrier that we have talked about, it acts as a barrier to the movement of large molecules from the blood into the tubules like cells. Okay, not these small glucose molecules, small proteins and all. Because these T helper cells are huge cells, right? So they will be not letting the cells pass by or pass through. They'll only let the smaller fragments, the smaller nutrients which can be passed. Okay, so that will be available for its exchange. Okay, so that is how the basal membrane, you can say, integrity is maintained. And finally, the waste products which are produced by the cells of the tubules, because biochemistry is going on, we'll be having Krebs cycle, we'll be having glycolysis, gluconeogenesis, glycogenolysis, then you will be having, you can say, nucleic acid catabolism, anabolism, so many biochemical, you can say, reactions. So it will produce the waste products, the carbon dioxide also through the Krebs cycle. Now the waste products which are produced by the cells of the tubules, they pass through the basal membrane again by diffusion and are carried away by the small veins which are present in the interstitial spaces. Okay. Now these interstitial spaces, they also contain something known as the testicular interstitial cells, or you may be familiar with it. They are known by the name as, more renowned name is the Leydig cells. Okay, this interstitial spaces, they contain, you can say, the testicular interstitial cells which are commonly known by the name as Leydig cells, referred to as Leydig cells. Now they synthesize and secrete androgenic steroid hormones. Okay, androgenic hormones, which are, all these hormones are steroid in nature. Okay, steroid hormones. Precursor is cholesterol itself. Okay. Now, if you talk about an what is an androgen? So, in order for an androgen is a molecule that will stimulate the growth and the maintenance of male tissues. Okay, so we have several naturally occurring androgens in men, means they all bind to a single androgen receptor. Okay, which is present in the cytoplasm only. Okay, and when bound, it translocates to the nucleus and acts as a transcription factor to alter gene expression. Just like I told you, they have estrogen receptor when I was talking about the luteal phase, okay, and the follicular phase, that why and how does the GnRH, okay, neurons, they recognize the higher and lower level, okay, of the estrogen, and how in the ovulatory phase it becomes a positive feedback loop, in the follicular phase it is a negative feedback loop. So we have talked about kiss, you can say, the kiss peptine theory and the arcuate and the paraventricular nucleus theory and all. The same way, what you have is, we have the specific androgen binding receptor also, which are present in the cytosol, and upon it, when it gets activated, means suppose this is a cell, okay, this is the nucleus, this is the DNA. Okay, so when the androgen enters, okay, because it's a steroid hormone, it will enter through the lipid bilayer. Okay, lipid bilayer, cell membrane, that is, it will be able to pass through it, and it will bind the receptor protein, okay, present here, and then it would migrate into the nucleus, and this complex will act as a transcription factor which will recruit RNA polymerase to that specific promoter and cause the transcription of that specific gene. That specific mRNA will come out, okay, and then that specific protein will be synthesized, okay. So this is the way that testosterone and all, they are the androgens, DHT, testosterone, dihydro, you can say, estenone. The what they do is they cause the development of the or the perpetuation of sperm formation, the secondary male sexual development. So that is how they cause the development. Same for the estrogen as well. Okay. So in an adult, okay, if the levels of steroid hormones in the blood are measured, okay, I'll tell you how many or what in what concentration the androgens are found, okay. So that will be micrograms per 100 liter of blood plasma, okay. First of all, it will be in microliters, just for your knowledge, okay, since you want to become doctors, per 100 ml of blood plasma, means I've taken out the RBCs, okay, but clotting factors remain, still plasma, not serum, okay. So testosterone, it is 0.7, okay. If I talk about testosterone, which is the main androgen, 0.7 means 0.7, you can say, micrograms, not microliter, it will be microgram here. Means 0.7 microgram per 100 ml of blood plasma. That is the concentration of testosterone. If I talk about 5 alpha dihydrotestosterone, okay, or simply I write DHT, it will be 0.05 μm per 100 ml, okay, microgram, means 10 to the power minus 6 gram, right? Then if I write here androstenedione, okay, androstenedione, okay, so that will be 0.1 only. These are all testosterone like ones, but they have difference in the function, okay. So when you reach the doctor level, you will know the difference in the function, or if we discuss somewhat later down the lane, I'll explain to you their functions as well. And one more is, if you have one more known as dihydroepiandrosterone, known as DHEA, okay, dihydro, at least there are four, okay, epiandrosterone. So its concentration is 0.5, okay, it's known as DHEA, 0.5 μm per ml. So just to give a perspective, these are the four types of androgen present in your, like in any male, like it is present in your body as well. In females also it is present, even HCG, prolactin, HPL is also present in male. It is not like those hormones are only female exclusive, but the concentration in your body will be much lesser in concentration when we talk about testosterone, but it is present in, you can say, females also. Just like I also contain estrogen, progesterone, the HPL, the HCG, the prolactin. I also contain those hormones, okay, because the genes are same. If you check, all the genes are same, but the expression level is different. So get that concept correct. It's not that I do not have the genes for progesterone. Thus that its expression level is much lesser due to again epigenetic regulation, right? Methylation, acetylation. So keep that concept in your head. Okay, it's not like the males, they do not have, you can say, HPL. From where those genes come from, right? So genes are the same. The DNA is the same, right? Now testosterone and DHT, that is 5 alpha dihydrotestosterone, they are the potent androgens, meaning that they are they stimulate the androgen dependent structures in very, you can say, low dosages. Okay, so about 95% of the testosterone, it comes from the Leydig cells of the testis, and the remainder comes from the adrenal gland. Okay, the zona reticularis layer of the adrenal cortex. Okay, to be very precise. So 95% of testosterone comes from the, you can say, Leydig cells itself. The 5% comes from the adrenal cortex. Okay, zona reticularis. So you have zona glomerulosa, zona fasciculata, and then zona reticularis in the adrenal cortex, in the cortical layer, and then you have the adrenal medulla. Okay, so that is again class 11th stuff from the endocrinology or the chemical coordination chapter, the last chapter, right? Now, approximately 98% of the circulating testosterone in any man is bound to the plasma proteins. Why? Because just imagine, what did I tell you about testosterone? It is a steroid hormone. Okay, get the concept. People are not aware of these concepts. They just talk about steroid hormones, but they're not aware of the concept. Even the thyroid hormone, let me tell you, is a steroid hormone. There is thyroid binding globulin, which will bind to the thyroid hormone in order to carry it in the blood, because blood is 70% water. So you cannot just make oil go through the blood. It will cause thrombus or it will cause a clotting. Okay. Suppose you took oil and there were no cholesterol particles. Those cholesterol particles were not, you can say, embedded in the proteins, because what are cholesterol? Cholesterol are same HDL, LDL, they are just difference in density. Okay, just know this for a fact. Only what happens is those lipids are, or lipid soluble molecules are coated by proteins. Those are known as apolipoproteins, like ApoB100, ApoA1. Okay, so these are the proteins that coat the cholesterol particles, and then they are known as HDL, LDL, VLDL, chylomicrons. Okay, IDL. Okay, intermediate density lipoprotein. That is why the full form is high density lipoprotein. What is lipoprotein here? Lipo means lipid plus protein, meaning that the lipid part, cholesterol or cholesterol esters, are embedded inside. They are embedded in the protein, and the protein, it is exposed to the blood and then it is carried. Otherwise, whatever oil you put in your body, in your what is called system, in your food, if there were no apolipoproteins, means apolipoprotein means the proteins which are not combined with lipoprotein, with the lipid, then with apolipoprotein and lipid, then it becomes lipoprotein, means combined. So if there are no apolipoproteins, those specific oil particles would have caused, you can say, thrombus or clotting in the blood, because they would be insoluble and they would obstruct the blood flow. Okay, so same way, thyroid hormone cannot pass through the blood. It is steroid hormone, lipid soluble, water insoluble. Same way, testosterone cannot pass through the blood. Okay, it is insoluble in blood. Same way as your benzene is not, you can say, soluble in water. Petrol is not soluble in water. Okay, so blood is a polar compound, right? So 98% of testosterone, okay, in males is bound to plasma proteins, okay, like sex hormone binding globulin, or the most prominent or dominant protein in the blood, which is known as albumin. Okay, sex hormone binding globulin or albumin itself. Okay, because this is the most predominantly abundant protein present in the, or I should be saying globular protein, because it's not a fibrous protein like elastin or collagen present in the blood. right? So the remaining 2% is in, you can say, cell permeable form, so that doesn't make that much of a difference, but 98% is bound, okay, to the proteins, okay, very special or specific, you can say, demarcation. This is always know, same for the estrogens as well, okay, always know this difference, okay. And the other ones like androstenedione and DHEA, okay, are not that potent. They are weak androgens, okay. They are produced mainly by the adrenal glands, not the Leydig cells, okay. And in research, their role in the male reproductive function is still not clear, okay. That their, what kind of role do they play, okay. They have certain roles, but that is beyond your course. But the real function as to the, or with regards to the human reproductive system, is still not or has not been elucidated in detail till now in the research, okay. So I hope you are able to comprehend today's lecture. If you have any kind of doubts or queries, you can definitely contact me on my personal number. I'll just clarify your doubts as soon as possible. Okay then. Thanks a lot, and let's continue this discussion in the